Hello, everyone. My name is Pradeep Singh Chauhan. I am a staff scientist in the laboratory of Dr. Adil Chaudhary at Department of Radiation Oncology from Washington. First of all, thank you to the CGC organizer for giving me opportunity to present the exciting result on our project, whose title is Combination of Genomic and Epigenomic Analysis of Plasma Cell-Free DNA identifies stemness features associated with the worst prognosis in high-risk metastatic castration-resistant prostate cancers. As you all know, prostate cancer is the second leading cause of death among US in men and also in worldwide. Though the localized prostate cancer can be treated effectively with the surgical resection or radiotherapy. However, the metastatic prostate cancers, they poses a clinical challenge and they account for the significant morbidity. The first-line treatment for the metastatic prostate cancer is androgen deprivation therapy. But eventually, all the prostate cancer patients, they progress to a lethal form of the disease, which is known as the castration-resistant prostate cancer. And these patients, they develop the AR resistance based on the mutations, aberrant modifications, and cofactor perturbations in the AR and associated cofactors. These patients, although they develop resistance, there have been past like significant PFS and OS has been improved with the advent of AR-directed therapy such as abiraterone and enzalutamide. But still, 30 to 40% of these patients, they developed AR resistance to the treatment. And so we thought that we need a molecular biomarker which can predict the resistance to these treatment, especially primary resistance, which can enable the clinician to think or consider the alternative treatment, such as radiotherapy or immunotherapy. So to address this issue, our collaborators, Kugle and Deng et al. In Cell 2018 published a paper in which they performed the whole genome sequencing and whole transcriptomic analysis of 101 tumor tissues. And they've observed that they found an enhanced amplification of a distal region enhancer, which is 624 kb upstream of the AR gene body. And they correlate it with the AR expressions. And interestingly, in this study, they found that 81% of the treatment-resistant cases, they have an amplification of these AR enhancer regions, and more importantly, it was present in 11% more than the gene body alterations. So with this, we try to use or leverage the circulating tumor DNA to detect all these clinical genomic alterations from the plasma cfDNA of metastatic prostate cancer patients. In this regard, we published a paper in JCPO 2022, where we devised a novel cell-free DNA assay called mCRPC. This is based on the CAPSIC platform, and we applied this assay to the 40 plasma samples obtained from the metastatic prostate cancer, and we found that AR and AR enhancer alterations, they were the most common somatic mutations observed in the 45% of AR-directed treatment-resistant cases. And moreover, these AR locus alterations, they predicted the resistance with high sensitivity of 78% and the specificity of 100%. And so after establishing the sensitivity and specificity in detecting the resistance of these resistance to the AR-directed therapy, we asked whether the mCRPC analysis can risk stratify these patients. So we showed that the patient with AR enhancer alterations, they have significantly worse PFS and worse OS associated with them. To further extend this mCRPC assay to the earlier therapy, so we tried to validate our study to detect the AR enhancer alterations in the plasma cell-free DNA of mCRPC patients who are going to receive the first line of AR-directed therapy. And for this, we enrolled 99 mCRPC patients from 2 institutions with the help of our collaborators, Dr. Oliver Sartor from Mayo Clinic and Dr. Russell Paczynski from WashU. And among these 99 patients, 63 patients, they received 63 patients have plasma samples collected just before the AR treatment. And we found that the pretreatment detection of AR locus alterations, they predicted the worse PFS and worse OS, which suggested that an mCRC assay can risk stratify the mCRPC patients. Next, we asked, can we learn more about the underlying biology of the high-risk mCRPC patient by studying the cfDNA epigenomic? For this, we performed the whole genome EM-seq, enzymatic methylation sequencing, on the 43 plasma samples collected before the starting of AR-directed therapy. And we found that these patients, And we found that the high-risk high-risk mCRPC patients, they have lower promoter methylation rate compared to the low-risk mCRPC. And these are the top 50 hypermethylated DMRs present in these high-risk mCRPC patients. And then we asked, can we infer the epigenomic or transcriptomic features from the cfDNA fragmentomic profiling? As you know, the cell-free DNA are the fragments that are released into the blood, and they are 167 base pair in length, protected by the nucleosomes. And this is an important observation because the nucleosomes, we can infer the nucleosome footprints from the cell-free DNA. And based on this hypothesis, in 2016, Peter Ulls published a paper in Nature Genetics and Snyder et al. In Cell 2016, and they hypothesizes that nucleosome profiling at the promoter site from the whole genome sequencing of plasma cell-free DNA can be associated with the expression of the genes. And similar to this hypothesis, Gavin Haw et al. From Fred Hutchinson, they published a paper in Nature Communication 2022, where they developed a tool called Griffin, which can be used for the nucleosome profiling of the cell-free DNA. So we used this Griffin tool to identify the transcription factor associated with our high-risk mCRPC patients. And we found that there are some transcription factors, such as GRHL2, which is a cancer-specific transcription factor, found to be highly active in high-risk mCRPC patient, as compared to another transcription factor, AMICOM, which is a blood-specific transcription factor, found to be active in the low-risk mCRPC patients. To further confirm whether these differential accessible transcription factors acquired on the plasma cfDNA, they showed the same result on the tumor also. So we found that the more accessible transcription factors in high-risk mCRPC patients, they were significantly upregulated in tumor tissue of localized prostate cancer cases compared to the least accessible transcription factor in high-risk mCRPC. Now, to further to gain insight into the pathways associated with these top 20 transcription factor more accessible, we performed the gene set enrichment analysis, and we found that these pathways were significantly enriched with the stemness and the differentiations. Now, next we asked whether we can infer the stemness signatures from the liquid biopsy. And for this, we used a tool called Cytotrace. This is a computational tool developed by the Gulati et al. From Dr. Aaron Newman lab at Stanford. Basically, this tool gives us a stemness score based on the differential capability of the cells. And each cell is assigned as a stemness score. And being high score means more stem-like, and being low score means less stem-like cell. So we use this Cytotrace on a published data set of single RNA-seq obtained from the 14 mCRPC patient, and we predicted the more stem-like genes and less stem-like genes, and developed a stemness-based scores which can risk stratify and predict the survival analysis in mCRPC. Using this tool, we were able to identify the gene enriched in more stem-like cells and also genes enriched in more stem-like cells. And since these stem-like signatures, they were obtained from the single mCRPC data, we also compared their methylation level in our cfDNA data. And we found that the high-risk mCRPC patients, they were enriched with the more stem-like cells. Conversely, we obtained that low-risk mCRPC patients were enriched with the low stem-like signatures. And after determining this, we also thought, like, can these signatures predict the prognosis of these patients? So we found that the patient with more stem-like signatures, more stem-like signatures, they were associated with the worse PFS and OS tending towards significance. To validate this finding, we used an external dataset of bulk RNA-seq obtained from the 80 mCRPC patient, and inconsistent with our results, we found that these patients with more stem-like signatures were associated with the worse overall survival. To conclude my study, plasma cell-free DNA alterations in AR and enhancer locus correlate significantly with the worse outcomes in mCRPC patients. Transcription profiles of mCRPC can be predicted from cell-free DNA epigenomics. High-risk mCRPC patients, they have more stem-like signature profiles, which correlate with the worst survival outcome. And last, it will be important to independently validate these findings with outside cohort and perform cfDNA tumor cross-correlative analysis. With that, I acknowledge my colleagues and my collaborators, and also the patient and their families. Thank you for your kind attention.